Good morning, everyone. I'm delighted to welcome you all today. Thank you for joining this session. Today, we will be exploring the validation of 70% isopropyl alcohol disinfection in cleanrooms. This presentation, led by Dr. S.K. Singh, Ph.D., will cover the key aspects of disinfectant effectiveness and validation processes. We'll look at the methodology, results, and implications for maintaining sterile environments. So, without further ado, let's dive into the topic and see how this important disinfection process is validated and optimized. Let's start by emphasizing that cleanrooms are critical environments where maintaining sterility is absolutely essential. To achieve this, we need validated disinfection programs that ensure consistent and reliable results. Now, one of the most widely used disinfectants in these settings is 70% isopropyl alcohol, or IPA. It's popular because it's effective for several key tasks: surface disinfection, cleaning equipment, and even sanitizing gloves. What makes IPA the preferred choice? Well, mainly its ability to kill microbes efficiently, which is critical in maintaining a sterile environment. Plus, it evaporates quickly, so it doesn't leave residues that could interfere with processes or product integrity. Its low residue also means it's less likely to contaminate surfaces or products, making it a trusted agent in these sensitive areas. Overall, choosing the right disinfectant like IPA supports our goal of maintaining high sterility assurance in the cleanroom. It's all about balancing effectiveness, safety, and efficiency to keep our environments safe and compliant. So, why do we use 70% IPA for disinfection? Well, the key lies in its mechanism of action, which involves both protein denaturation and membrane disruption. Essentially, the alcohol breaks down vital proteins in microorganisms and destroys their cell membranes, leading to effective sterilization. 70% IPA is considered optimal because the water content actually helps improve penetration into the cells. It allows the alcohol to diffuse more effectively through the microbial cell walls. Plus, since 70% IPA evaporates more slowly than higher concentrations, it provides better contact time on surfaces, making it more effective overall. This concentration works well against a variety of pathogens, including vegetative bacteria, fungi, and enveloped viruses. These organisms are generally less resistant, so this concentration hits them hard. However, it's important to recognize the limitation here. 70% IPA is not effective against spores. Spores are much more resistant and require different disinfectant strategies. Understanding these points helps us appreciate why 70% IPA is a go-to disinfectant in many settings. It's a balanced formulation that maximizes microbial kill while maintaining practicality in application. Let's take a closer look at the key regulatory requirements that guide our disinfection practices. First, we have the EU GMP Annex 1 from 2023, which sets out the standards for sterile manufacturing and emphasizes the importance of validated disinfection processes. Then, there's USB 1072, which provides guidance specifically on disinfectants, ensuring their proper selection and use. The WHO TRS 961, particularly Annex 6, also plays a crucial role by outlining international standards for contamination control, including disinfection procedures. And we can't forget ISO 14644, which focuses on cleanroom standards and controlling airborne contamination. The core requirement across all these guidelines is that our disinfection process must be validated for both effectiveness and the specific use conditions. This means we need to prove that our methods reliably eliminate contaminants under the actual conditions in which we operate. Validating our disinfection process isn't just about compliance. It's about ensuring safety, quality, and consistency in our manufacturing environment. It ties directly into the overall theme of maintaining a controlled, compliant, and safe production space. Let's focus on the validation objective for this slide. Our goal here is to demonstrate that the 70% IPA solution is truly effective. First, we want to show that it reduces microbial load effectively, which is critical in maintaining a sterile environment. It's important to note that this solution works on all cleanroom surfaces, ensuring broad applicability across different areas. Additionally, we need to confirm that it remains effective under actual usage conditions, not just in controlled lab settings. To ensure consistent results, we also need to establish key factors, such as the contact time, the amount of time the solution needs to stay on a surface to work effectively. We must determine the appropriate frequency of application to maintain cleanliness and identify the most effective application method to maximize coverage and efficacy. By thoroughly validating these points, we can confidently affirm that our disinfectant performs reliably in real-world cleanroom environments, supporting our overall goal of ensuring safety and sterility. Let's walk through the validation approach. Typically, this involves a study design where we start by surface inoculation with known organisms. Then, we apply 70% IPA, making sure to control the contact time exposure. After that, we recover the microorganisms and calculate the log reduction to assess how effective the disinfectant is. The test organisms we focus on include Staphylococcus aureus, Pseudomonas aeruginosa, and Bacillus subtilis to check for spores. This helps us understand the disinfectant's efficacy against both bacteria and spores. Now, the scope of validation is quite comprehensive. It must cover different surface types like stainless steel, epoxy floors, glass, and plastic, because these are common in many environments. It also needs to account for various cleanroom grades, from ISO 5, which is grade A, to ISO 7 and 8, which are grade C or D. In terms of application methods, we consider both spray plus wipe techniques and mopping. This ensures that our validation reflects real-world cleaning practices in different settings. Overall, this approach ensures that our disinfectant performs reliably across various surfaces and cleanroom conditions, matching the overall goal of maintaining strict hygiene standards. Let's take a closer look at the acceptance criteria, as outlined by USP 1072. The baseline requirement is a minimum 3 log reduction, which equates to a 99.9% reduction in bacteria. This means that our product must significantly reduce microbial presence to meet safety standards. For critical areas, the expectations are even higher. Specifically, for grade A and B environments, we're aiming for a reduction of 3 to 5 logs. That's a 99.9% to 99.999% reduction, emphasizing the importance of microbial control in these sensitive zones. Additionally, it's essential that there is no microbial increase during the shelf life study. This ensures that the product remains safe and effective throughout its intended storage period. Now, let's briefly look at how we calculate this log reduction. The formula is straightforward. Log reduction equals the log base 10 of the initial count divided by the final count. For example, if we start with a bacterial count of 10 to the 6th power, or 1 million bacteria, and end with 10 squared, or 100 bacteria, then the calculation looks like this. Log 10 of 106 divided by 102, which simplifies to log 10 of 104, that equals 4 logs of reduction. In this case, a 4-log reduction meets the acceptance criteria, demonstrating a substantial decrease in microbial load. Understanding this calculation helps us verify that our sterilization or decontamination processes are effective and compliant with USP standards. All right, let's look at this surface challenge study example. Initially, we started with 1 million colony-forming units, or CFUs, applied directly to the surface. This gives us a baseline of how much contamination was present before any treatment. After applying the IPA, or isopropyl alcohol, we recovered only about 100 CFUs. This significant reduction indicates that the disinfection process was quite effective. So what does this tell us? Well, the key point here is that the IPA didn't just reduce the contamination, it demonstrated effective disinfection. It shows that the surface was thoroughly cleaned and sanitized, which is crucial in maintaining hygiene and preventing the spread of microbes. This example ties into the overall theme of our presentation, highlighting the importance of proper disinfection techniques and their proven effectiveness in real-world scenarios. In this slide, we're looking at the in-use stability or hold time study, which is crucial for understanding how long our prepared 70% IPA remains effective on surfaces. The procedure involves storing the prepared IPA and testing it at various time points, specifically at 0 hours, 24 hours, 48 hours, 72 hours, and 96 hours. The key here is to ensure that there is no increase in microbial growth over these time intervals, which confirms the stability and effectiveness of the solution during use. We also need to establish an expiry time, which could be, for example, 7 days or 24 hours, depending on the results. This helps define how long the IPA can be reliably used after preparation. A critical part of this study is contact time validation. Basically, we need to make sure that the IPA remains wet on the surface for a sufficient period, typically between 30 seconds and 1 minute, to ensure proper disinfection. The study compares immediate drying versus wet exposure, highlighting that maintaining a wet surface during this contact time is essential. The main takeaway here is that wet contact time is a vital factor for the efficacy of the disinfectant. If the surface dries too quickly, it may reduce the disinfection effectiveness, which is why understanding and validating this contact time is so important for reliable surface disinfection. Now, let's talk about some limitations and how we can adopt a risk-based approach. First off, this method is not sporicidal, meaning it doesn't effectively kill spores. So, if spores are a concern, we need to consider additional measures. Also, evaporation can reduce the efficacy of the solution over time, which is something to keep in mind during application. Another point is that sprays can generate particles, and this might pose a risk, especially in terms of aerosolization or inhalation. To address these limitations, we can implement some mitigation strategies. For example, rotating the use of different agents, like hydrogen peroxide or peracetic acid, can help enhance effectiveness and reduce resistance. Also, using a controlled spraying technique is crucial. This means applying the solution carefully to minimize particle generation and ensure proper coverage. Overall, understanding these limitations and corresponding mitigation tactics helps us adopt a more effective, safe, and risk-aware approach in our disinfection protocols. Let's talk about documentation and GMP compliance. First, there are several key documents we need to have in place. These include the validation protocol and report, which ensure that our processes are thoroughly tested and validated. We also need standard operating procedures, or SOPs, for the preparation and use of materials to guarantee consistency and quality. Additionally, a disinfectant rotation program is essential to prevent microbial resistance and maintain a clean environment. Training records are vital to demonstrate that staff are properly trained and competent. Lastly, batch traceability allows us to track each batch through every stage, ensuring accountability and quick identification of issues if they arise. Now, from a GMP perspective, all these documents and practices are not just paperwork. They are part of our broader contamination control strategy, or CCS. They help us maintain a controlled environment and prevent contamination, which is the core of GMP compliance. Remember, documentation isn't just about compliance. It's about creating a reliable, safe process that supports the overall quality of our products. So, wrapping up, I want to emphasize that 70% IPA is a validated standard disinfectant used in cleanrooms. This means it's proven to be effective and reliable for maintaining sterile environments. When validating its use, it's crucial to demonstrate a few key points. First, its efficacy in eliminating microbes. Second, that it's compatible with the surfaces we're disinfecting. And third, that it performs well under real-use conditions, just like in everyday operations. Ensuring compliance with these validation steps is essential because it helps guarantee product sterility, which is vital for safety and quality. It also supports regulatory approval, making sure we meet industry standards and helps us stay audit-ready by having documented proof of proper disinfection procedures. Overall, the goal is to maintain a sterile, compliant environment that supports the integrity of our products and the trust of those who rely on them. For further support, please feel free to contact us. We're here to help you implement what you've learned today and answer any questions you might have. Remember, your success is our priority. Thank you so much for your attention and participation. We appreciate your time and look forward to working with you.